It is my distinct pleasure to introduce today's moderator, Dr. Emily Binard. Emily earned her PhD in molecular and cellular biology at the Institute of Functional Genomics at University of Montpellier in France, where she investigated DNA replication origins and chromatin structure and senescence. She then went on to pursue postdoctoral research at the Gladstone Institutes affiliated with UC San Francisco, and the Buck Institute for Research on Aging, also in California, where she focused on HIV latency, aging, and mTOR signaling. She then transitioned to industry, holding scientist roles at BioAge Labs and Durion Therapeutics, where she advanced to principal scientist before working in her current position as senior scientist at Altos Labs. In addition, she's also been an early reviewer and editor at Bioprotocol. And with that, the floor is all yours.
Thank you, Renate, for the introduction. It's my distinct pleasure to introduce the webinar speaker of today, Michael Meadow. Michael is a medical student at the University of Rochester School of Medicine and Dentistry, where he recently completed his doctoral research at the intersection of proteostasis and disease. He conducted his PhD work in the labs of Vera Gorbunova and Andrei Selyanov, renowned for studies on aging and genome stability, as well as in the lab of Sina Gamagami, which specializes in systems-level analysis of protein turnover and degradation. During his undergrad study at UCLA in microbiology, immunology, and molecular genetics, Michael gained extensive experience in proteomics and neurobiology, including work at the UCLA Proteome Research Center. His research and publication record span multiple fields, including stem cell biology, neurodegeneration, and public health. Notable contributions include studies on iPSC-derived glial progenitors, autophagy lysosome defects in inherited disorders, and behavioral interventions to increase organ donor registration rates. Michael's academic excellence has been recognized with several honors, including the Jack and Faye Natchin Medical Student Summer Fellowship and a Medical Student Research Scholarship. It's my great pleasure to have you, Michael Meadow, to talk about your protocol and this very exciting technology. Please take the floor.
Thank you so much. Just pulling up the slides. Okay. So thank you so much, Dr. Besnard and Renee, for that introduction, as well as Bioprotocol for working with us in publishing our protocol, but also hosting this bio webinar today. It is my distinct pleasure to be here and share with you an exciting methodology we've developed that we call proteome birthdating. That was an integral part of my thesis, which I defended recently in the labs of Vera Gorbunova and Sina Gamagami. And yeah, so today I'm going to tell you a little bit about birth dating and how it is a single sample approach for measuring global protein turnover and this kind of new area of protein age. And so just to start out, this is our bioprotocol that was published in May of this year. And I would encourage everyone to check it out. It has a lot of tips and tricks as to how we did the experiments, but also how you can adapt the workflow to your own in vitro system or protein or pathway of your choosing. And so I'm going to start out today by talking about and introducing the idea of protein lifetimes, as well as talk about bottom-up proteomics. Transition to talking about how we innovated upon current methods to kind of develop proteome birth dating for measuring global protein turnover, and subsequently how we can use it to get at this idea of possible age selectivity or age intrinsic properties of proteins, including ubiquitination. And finally, I know a lot of people are very curious as to kind of how this method can be used going forward, maybe in the fields of neurodegeneration and cancer. And so I'll talk about a little bit about where we're going with it, and also maybe for some ideas as to where others can take this as well. So starting out on the most fundamental level, proteins are comprised of amino acids, and these amino acids are continuously being synthesized by ribosomes into their protein. These proteins go on to live their own molecular lifetimes in which they carry out the myriad functions we know to be ubiquitous of proteins and cellular functioning. But eventually, all of these proteins that are successfully synthesized will become degraded and recycled back into their constituent amino acids. And so one kind of corollary that kind of emerges from this is the idea that each of these proteins that has been successfully synthesized actually has its own molecular lifetime. And so if we look down here, we can see a protein being newly translated coming out of the ribosome before it lives its lifetime for some amount of time. During this time, it can accumulate damage, be repaired, be modified, and of course carry out its function. But again, eventually it will become degraded. And so a couple of important factors play a role in governing how fast the protein may turn over. And I'll start by talking about intrinsic properties of proteins. So the first and foremost, I would argue, is that protein function is often intimately linked with protein turnover rate. So for example, transcription factors, which need to be rapidly responsive to their environment, are some of the shortest lived proteins in our cells, whereas whereas housekeeping proteins, maybe like FDH, are some of the most long-lived in our cells. Other properties of proteins that can contribute to this turnover rate could be their structure, and with this, their stability and their propensity to remain folded, their aggregation state or their aggregation chronicity, as well as sequence determinants, for example, degrons or the end rule, and looking at the N-terminal amino acid, as well as modifications and even where the protein is in the cell. There are some extrinsic and environmental factors as well. So for example, the temperature will influence how fast these things happen. The intrinsic activity of, say, of the pathways responsible for this degradation would also play a role, as well as the species, and even within the species, the tissue of origin is known to play a role in these protein turnover rates. And how this has been studied historically has really been through very historically through the use of radioactive methionine incorporation, or kind of like a chase experiment where you see how fast it disappears, and using kind of radiography to measure this, or other probes. The field kind of transitioned into really using Western blotting, as well as kind of cyclohexamide, which is a translation inhibitor, or degradation inhibitors like MG132 to track protein abundance. And now we're kind of in an era where a lot of kind of global turnover studies use what's known as dynamic SILAC, or stable isotope labeling by amino acids in cell culture. And we'll really be focused here today on D-SILAC, and I will go into that into more detail throughout the talk. But it hinges on the use of bottom-up proteomics. So the way that we study proteins in their lifetimes is through bottom-up proteomics, in which we can take a complex mixture of proteins or cells, first lyse those, and subsequently take those proteins and digest them with a protease called trypsin. And we can inject these fragments into a machine seen, or an instrument seen here. This is our LC-MS Astral that we have here at the proteomics core at the University of Rochester. And so when we take these fragments and we inject them into this instrument, what we're able to do is quantify, identify, and even identify modifications on thousands of peptides throughout the proteome. And moreover, we can map those peptides back to their originating protein. And we can do this for more than 5,000 proteins throughout the proteome. And you can do this for any model organism as well. And the kind of approach that we take to using bottom-up proteomics and studying protein turnover is in the context of aging. And we do this because, and so here I'm just showing the 12 molecular hallmarks of aging on kind of a gross scale. We're all familiar with what an aging person or animal might look like. But on a molecular scale, a lot of work has been put into defining these 12 kind of hallmarks of aging. I would argue maybe the most important of which is this loss of proteostasis. So the loss of health of the proteome at large, as well as the ability to kind of repair it. And so with this loss of proteostasis, it is thought that this kind of contributes throughout the aging process. It contributes to the development of age-related proteinopathies. Maybe one that we would be most familiar with, unfortunately, could be Alzheimer's disease, where if we look at kind of the parenchyma of the brain, you can see these large brown deposits of A beta plaques. And as we know, Alzheimer's disease is a truly devastating proteinopathy. Wherein there remains no effective cure or even really treatment to slow progression of these symptoms, but also of the molecular biology and the protein aggregates. And so the complementary approach we take to aging and protein turnover is one of comparative biology. So mammals as a class span more than 100-fold in terms of their maximum lifespan. And I'm showing here just a selection of 12 or 13 of these animals with their corresponding maximum lifespan. And you can see actually that long life has evolved multiple times on multiple different lineages of this tree. And that suggests that longevity has evolved multiple times in mammals and possibly through different mechanisms. So we use this kind of approach to elucidate some of these mechanisms, one of which could be proteostasis and protein-based. And so a previous student in the lab, who I hope is here today, named Kyle Swovic, took skin cells from these animals. So we have short-lived, long-lived, medium-lived. And he did what was known as D-Silac, which I introduced a little bit earlier, in which he took these cells, grew them to confluence, and took different plates and labeled them for different amounts of time with a heavy labeled lysine and arginine. And using this, what he found out was that actually longer-lived species, like for example, the bowhead whale, actually turned over their proteins as a whole much, much slower than shorter-lived species like the mouse. And this is actually somewhat counterintuitive. This is because, for example, or one would think that maybe these longer-lived species were more efficacious at degradation as a whole, as, for example, the bowhead whale, their neurons, right, post-mitotic cells are really dependent on kind of maintenance of the proteome and don't have a lot of proliferation potential or repair potential. And so if we look at the bowhead whale, you have a species that has a brain that lives for more than 200 years. And so you would really think that maybe these bowhead whales would be much better at protein degradation than the mouse or these shorter-lived species. One kind of hypothesis that emerges from this is that these longer-lived species may be more efficient or selective at degrading the proteins that necessitate it. One basis of this kind of bias could be that of age selectivity, and I will get into what that means shortly. Okay, so I've introduced the idea of protein turnover, kind of the significance of it in terms of aging, and also introduced kind of how the current methodologies used to study it. And so now I'll talk about where the field is at right now in terms of dynamic SciLac and how we innovated on it through the development of proteome birth dating to study global protein turnover. And so our bottom-up workflow for proteomics, and this will be the same for both dynamic SciLac and proteome birth dating, is we take our plate of cells and we perform a typical lysis and protein extraction. With these proteins in hand, we can then perform a tryptic digestion, as well as reduction and alkylation of these peptides. And we can take this mixture then, perform our tandem mass spectrometry. And from this, we can end up with our mass spectra. Our mass spectra can then be bioinformatically manipulated using first, we'll use FragPipe to identify and quantify the peptides, as well as map them back to their original proteins. And lastly, we use in-house scripts, but you don't have to. But we use Mathematica to then process the data and come up with some meaningful biological insights. And all of our scripts that I will talk about are all publicly available online already. And if you need easy access to them, they should be in the bioprotocol. So I'll start first by talking about more specifically dynamic SciLac. So again, this has been the kind of gold standard in the field for about 10 to 15 years now. And in this methodology, you start with a plate of unlabeled quiescent cells. And if we were to do mass spec on this, you would see that you just see native proteins, essentially. You have your one peak as well as some additional kind of just environmental isotopes. We can take another plate of the same cells then and culture it in media containing a labeled amino acid, so a lysine or arginine with a few extra neutrons on it. And doing this for some amount of time, some of the proteins will incorporate this heavier label. We can do the same thing again for another plate of cells for a different amount of time with the same amino acid. And again, for another plate of cells, a different amount of time with the same amino acid. And seeing how much of this label is being incorporated into your proteins during any given interval allows you to kind of, if you plot the amount of each protein labeled or the fraction labeled of each protein over this period of time, you can use a first order equation to determine kind of the half-life or rate of turnover of a given protein or peptide. In this case, we have a theoretical, actually a practical peptide that measures a half-life of about 2.3 days. And so this is a very good technique. It has fostered a lot of good science. However, there are some caveats. So maybe one thing you notice is that these are all of these individual samples that comprise these points are coming from different plates of cells. And so this introduces some amount of experimental variability, some amount of difficulty being able to control and normalize, as well as it can get quite expensive, both in terms of the sheer growing of the cells in the media, but also in terms of a lot of times these samples will be complex and each sample will require multiple mass spec runs. And so the costs really start to add up in that regard. We're also limited to just unlabeled, one label, and then you could add a third label if necessary, but you are limited to just 3. And so we devised a strategy that allows us to do this in one single plate of cells, as well as incorporate 5 different labels as opposed to just the standard 3. So our way of doing this was to take, again, just a plate of cells grown to quiescence and culture them in media containing regular unlabeled lysine. And we can switch the cell culture media for some amount of time to a media containing a lysine enriched with 4 extra neutrons. We can do this again with the same plate of cells for another amount of time with the next label. This label also has 4 additional neutrons, but notably, they are differentially rearranged in the molecule, and I'll get back to why that is important in a second. We can also do this then with another label of 8 neutrons added to this lysine, and the corresponding pair of that would be 8 neutrons with that rearrangement. And what that rearrangement allows us to do by mass spectrometry is if we look at the spectra, on low resolution, we see that we kind of see the standard 3 peaks of maybe a SciLac experiment. However, upon higher resolution, we can actually go in and tell these the very close in mass pairs apart, and we're able to detect and quantify all 5 of these labels incorporated in a given protein. And doing this for the same practical peptide that I showed on the dynamic SciLac slide, you can see that this peptide has accumulated some amount of each of these labels in a distinct distribution that we can then model and come to the same conclusion that its half-life is approximately 2.3 days. Yeah. And so the reason we're able to do this and use the 5 labels, as I mentioned, is this rearrangement of the heavy isotopes within the lysine molecules. And this is facilitated by our use of neutron-encoded amino acids, or nucode amino acids. So here I'm showing you the pair of +4 lysines that we were using. You can see that the isotopes are differentially rearranged within them. And actually, this slight rearrangement is enough to change the nuclear binding energies of these atoms and this molecule, such that it confers an ever so slight mass difference in the order of millidaltons. And so what that allows us to do is we don't actually add to the complexity of the spectrum at all on a low resolution scan. But again, on the high resolution scan, we are able to detect that ever so small mass difference conferred by the rearrangement of the isotopes. So putting this all together, if we start with a plate of unlabeled cells now, we culture it in our first lysine for 4 days, our second for 2 days, our third for 1 day, and our fourth for a quarter of a day. We end up with a plate of what we call birth-dated cells. And actually, all of the information within each experiment is available to or encoded in each individual cell, which also is a unique feature relative to dynamic SciLac. And we can take these birth-dated cells and calculate their corresponding half-lives. And so here I'm just showing 3 example proteins, one of short half-life, and you can see that it has incorporated mostly the most recent label, one of kind of a medium half-life, 3 days, and you can see that it correspondingly has incorporated kind of those middle labels, and one of a long half-life of 8 days, where you can see that most of this protein population consists of the first label or unlabeled lysine that was actually used before the start of the experiment. And so we can do this not only for that one peptide I showed you and these 3 example proteins, well, rather, we can do that for not only theoretical proteins and peptides, but we can actually go and do this. And so here I'm showing you one actual example of a known short-lived protein, rhombosbondin 1, and the corresponding amount of each label that has been incorporated into the protein. These faint lines represent peptide level measurements, and the solid dots represent protein level measurements, and they're just the median of the peptide level measurements. And you can see that corresponding to a short half-life, that rhombosbondin 1 has incorporated mostly, or what we can detect to be mostly, the most recent lysines of the experiment. We can also do this for long-lived proteins like aldehyde dehydrogenase. And so you can see here that correspondingly, it has accumulated mostly the eldest labels of the experiment. And that is consistent with its known long half-life. And we can do this not only for just these 2 proteins, but we can actually do this for more than 5,000 proteins throughout the proteome. And so this is a bit of a complicated plot, so I'll take some time to go through it. On this plot, each vertical line represents a unique protein, and those proteins have been rank ordered according to their known half-lives. On the y-axis is the log transformation of half-lives, so towards the bottom are shorter half-lives and more rapid turnover. Towards the top are longer half-lives and slower turnover. Each blue dot on a vertical line represents the peptide level measurement, and the green dots represent the median and the protein level measurement. And so what I'd like to point out is a couple of things. One, that we can, of course, do this for many, many, many proteins throughout the proteome, 5,000 in this plot. Next, our measurements align very well with published measurements. As I mentioned, these are rank ordered according to their known half-lives, not our calculated ones. And lastly, we can see that the peptide measurements for any given protein are pretty tight in their range, but also are very tight in their range around the median. and we can get very kind of reliable and accurate data from these. And so, of course, we wanted to compare between dynamic SciLac and proteome birth dating to see if we were being accurate and how the two kind of compare in their metrics. And so we did both dynamic SciLac and proteome birth dating in parallel on just human skin cell. And so here I'm showing kind of our statistics and data from our dynamic SciLac experiment. We had 4 different time points, and we were able to quantify the turnover rate of about 1,200 proteins throughout the proteome, with a pretty good evidence level of about like 8 PSMs per protein. But then when we compare that to our proteome birth dating experiment, we actually were able to quantify the half-lives of more than 5,000 proteins. I guess with quality control, maybe 4,000. And demonstrate that we're getting, in this case, better coverage than dynamic SciLac, but at least similar. And when we take our results and calculated half-lives from either of these experiments and we plot them on just a pairwise comparison, so on the y-axis, we have our half-lives resulting from dynamic SciLac. The x-axis, we have half-lives resulting from our proteome birth dating experiment. And you can see that they align really, or they agree with each other really quite nicely, and they're right along the line of true fit. So we can kind of say that we're actually doing quite well relative to dynamic SciLac methodologies. But how about kind of the methodology itself? How is it doing internally and relative to published values? So here, I'm just showing you the pairwise comparison of 2 different replicates of a proteome birth dating experiment. It's the same cell line, but the experiments were done at 2 different times. And you can very nicely see that the replicates, so these are essentially biological replicates, align very nicely with each other as well. And so we're getting reliable and reproducible data from this workflow. But does it actually biologically make sense? And is that meaningful? And so here we just took some of our favorite protein groups and protein groups that we knew would kind of have an associated trend in their half-lives. So again, we have like something like transcription factors, which are known to be short-lived proteins. And correspondingly, if we look at our calculated half-lives, we see that it is in fact a shorter-lived protein group or group of proteins. Conversely, as I mentioned, like GAPDH and more housekeeping proteins like those involved in glycolysis tend to have a longer half-life, and we do see that here as well. Okay. So I've shown you now how we've kind of innovated on dynamic SciLac to develop this proteome birth dating technique, which works well in terms of kind of your routine experimentation in studying global protein turnover, and in fact has quite a few advantages relative to dynamic SciLac. But now I'm going to tell you about, biologically speaking, why we're so excited about this methodology. And that is because the way we've designed the workflow, it actually allows us to get at this idea of a protein's age and exploring how a protein's age may impact its interactions with its environment. So as I mentioned at the beginning of the talk, each protein that is successfully synthesized will have its own molecular lifetime and eventually succumb to degradation. A little bit of or if it's easier to think about, one can kind of draw a comparison to humans. And so here you can see this baby protein initially being translated, maybe learning how to crawl, learning how to walk, run, maybe finding a nice dancing partner, and eventually passing away. And while we're unfortunately familiar with how that passing away happens on kind of a gross organismal level, how that happens on a molecular scale largely occurs due to a protein called ubiquitin. So ubiquitin is a small little protein which serves to tag other proteins for degradation, either when they are unwanted, damaged, aberrant, resource conservation, a whole bunch of factors could play into this. So largely what happens when a protein is ubiquitinated is that it will serve as a signal for proteasomal degradation of the substrate protein. And in doing so, the substrate will pass through the proteasome and be proteolyzed back into its constituent amino acids. Ubiquitin is also known to help send substrates to more autophagosomal pathways. However, this is kind of an emerging trend and will really be focused on ubiquitin's large role in directing substrates to the proteasome via the ubiquitin proteasome system. And so we're focused here because it is possible that ubiquitin represents a putative age-selective mark. And so what I'm showing here is the null hypothesis and also the kind of state, the current state of methods of the field. So if we were to ask the question, which of these proteins of different ages were to be ubiquitinated, currently, methodologies would not be able to tell the difference, and also it's possible the hypothesis is wrong, and any age can be ubiquitinated and degraded. And in this way, one can kind of, again, in the null hypothesis, see that the ubiquitinated proteome has no difference in age distribution relative to that of the proteome at large. However, it is well known that when proteins become damaged or misfolded and aggregate, that these proteins tend to become ubiquitinated and degraded. And all of these processes, the damage, the aggregating, the ubiquitination, these are all processes that are thought to take some amount of time. And in this case, one can imagine that maybe the ubiquitinome or the ubiquitinated proteome may actually be somewhat older than the proteome at large. There are also, however, well-characterized cases of what's known as ribosomal quality control. So this is when something like a collided ribosome or a co-translationally modified protein or a stalled ribosome recruits this ribosomal quality control complex, most notably containing LTM1 or listerin, and targets nascent proteins for degradation. In these cases, you can imagine that the ubiquitinome at large, the ubiquitinome might actually be a great deal younger than the rest of the proteome at large. And so what proteome birth dating allows us to do is take this blue molecular lifetime and make it a little colorful, but more specifically allows us to retroactively separate these proteins and partition them on the basis of age or their time of synthesis. And so again, we can compare this to humans. We can ask the question, which of these populations of proteins, if any, comprise the majority of what is being ubiquitinated and subsequently sent for degradation by the proteasome. So how we did this, we used our birth dating technique. So we started again with our plate of unlabeled cells grown to quiescence. And we subsequently put it through the 4-day, 2-day, 1-day, 6 hours. We end up with our plate of birth dated cells. And now you can kind of appreciate that depending on which of these lysines a protein has incorporated, that lysine will correspond to when it was synthesized or its age. So if a protein that we detect has the first lysine in it, that would suggest that that protein molecule is more than 7.25 days old. And so we can start to kind of age these proteins and separate them out. We can, again, and so doing this then, we can ask the question in our cell with all these birth dated proteins, are there any subpopulation of these proteins whose age distributions differ? In this case, we'll be talking about ubiquitination, but just in kind of a vague example, you could kind of imagine an unmodified protein would have maybe one age distribution, and the modified versions of this protein could have a different one, perhaps. And so we took our birth dated cells and we wanted to investigate the age selectivity of ubiquitination. And to do this, we added MG132 for the last 6 hours of the experiment. So coincident with that last label. And MG132 is a well-known and well-characterized proteasome inhibitor, otherwise known as bortezomib. And so we build up these polyubiquitinated substrates. And by virtue of our mass spec workflow, then the triptych digestion on any residue that was ubiquitinated actually leaves a characteristic diglycine remnant. And this remnant people have designed very well-characterized commercial antibodies for. And so we can pull them out. And we see something really interesting when we do that. So if we start with Vimentin with just a unique kind of age distribution of labels, and we overlay then the KGG peptides that we can detect from them, we can see that these KGG peptides are just ever so slightly older than the unmodified forms. However, that actually comprises the minority of cases. What we see in the majority of cases is actually something like DNPK1, where we have this age distribution of the unmodified peptides. And when we overlay the KGG peptides or the ubiquitinated peptides over that, you can see that they markedly differ in their age. And not only that, it's that the KGG peptides seem to be significantly and markedly younger than the unmodified forms of the protein. And as I mentioned, this is actually the vast majority of the trend that we see. So if we start again with this plot that represents each vertical line as an unmodified protein and peptide, we can then overlay our ubiquitinated marks on top of that. And so what I hope you can appreciate is that we have a lot of these in the screen box, a lot of ubiquitinated marks, and the vast majority of ubiquitinated marks, which are quite significantly younger than their steady state age. And again, so on the y-axis, we now have age, where young is toward the bottom and old is toward the top. And you can see that regardless of the steady state protein's age, the ubiquitination events that we're able to detect seem to be quite a bit younger or even nascent. And actually, these proteins and peptides build up more than 4-fold upon MG132 treatment. And we can contrast that with these older ubiquitinated marks that we can detect, which do not build up at all with MG132, and also are far less frequent, but somewhat older. And so the real story here is how young all of these ubiquitinated marks are. And we wanted to kind of dig into this bioinformatically. And so just a slightly different way of looking at this as a histogram, we have all of our ubiquitinated proteins that we can detect and all of our unmodified proteins that we can detect. And we can see that their median ages differ markedly at about 3 hours versus 3 days. And so bioinformatically, we then wanted to identify maybe protein groups or Go terms associated or understand the rules about this nascent degradation we were seeing. And so on the y-axis here, we're plotting kind of the degree to which, or the dramaticity of how young the KGG peptide was relative to its unmodified counterpart. And we can see that among the youngest ubiquitinations are proteins destined for the nucleoplasm and cytosol, or even the mitochondria. But importantly here, they're synthesized in the cytosol. And we can contrast this to proteins which are synthesized on the ER and destined for, say, the Golgi, where there is no ubiquitination that takes place in the ER lumen. Those proteins have to be retroactively removed from the lumen to be ubiquitinated and degraded. And so we think that that process as a whole takes a lot of time, and that could be one explanation for why we see this. We also see, though, that a lot of the proteins with the youngest trend seem to be those in large protein complexes. Now, this isn't true for all protein complexes, as you can see here, but it is true for a lot of them. And so putting these two bioinformatic findings together, we can kind of come up with one rule that seems to play a role in how pervasively nascent these proteins degradation is, in that these could be orphan subunits, cytosolic orphan subunits, which fail to incorporate into their large cytosolic complex and cognate complex, and are therefore ubiquitinated and degraded. And so, in summary, we've developed this birth dating technique to partition proteins based on their age. We strikingly found that the vast majority of proteins that seem to be ubiquitinated and destined for the proteasome seem to be among this youngest population. And these older guys, we're not really sure what's happening with them. They could be destined for the proteasome, but of course they could just be regulatory marks like a K63 ubiquitination, or they could be going down other degradation routes entirely, like autophagy or specific proteases. And so this has now been published a year and a half ago in Molecular and Cellular Proteomics, and I'm very excited to share it, both because I'm proud of it, but also because it really, we feel, opens up a lot of room for future science and open science, and using this technique both to study the age selectivity of ubiquitination in disease and health, but also we believe this kind of technique can be broadly applicable to other protein or one specific proteins of interest, other pathways of interest, other diseases of interest, and I would argue even other macromolecules of interest. And to this end, we have also published the lovely bioprotocol, which goes into extensive detail as to how the experiments were conducted and how you can birth date cells of your own for your own technique. And I would encourage you to visit the protocol if you choose to do so. And so I will just conclude by talking about some possible future applications and directions that you or me could take using this methodology. And so one could be getting at this idea or getting back to this neurodegenerative context and looking at what is the age of proteins that are actually involved in these large aggregates that are so problematic in aging. And so as I've been talking, I've probably really stressed that protein damage takes some amount of time. And so one could kind of imagine that maybe these older lived proteins are those that are aggregation prone. However, it is also certainly possible, and one hypothesis could be that given the pervasive extent of nascent degradation we see in just normal human cells, it could be that in aging or in certain diseases, there's a failure of nascent degradation, and maybe it is actually these undegraded nascent products which are contributing to aggregation formation or aggregate formation. We can also, to this end, look at protein damage itself and try to ascertain what the age of a damaged protein is. And so here I'm just using an example of protein oxidation. And so again, I've probably been talking as the protein would accumulate this oxidation over some amount of time. But you can also imagine that as a protein is unfolded and emerging from the ribosome, many internal residues like methionines could be exposed to ROS and oxygen molecules that it otherwise wouldn't be. And in this way, or rather, we can kind of ask the question, when does protein oxidation or damage tend to occur in a protein's life? And correspondingly, how does that contribute to flux through the proteasome? We can also just ask about the other degradation pathway in general, major degradation pathway in general, autophagy. And we kind of have a hypothesis that perhaps these younger proteins are so akin to the proteasome because they're partially unfolded and can kind of glide through this porous complex, whereas these larger, maybe older aggregates wouldn't be able to pass through as easily, possibly stalling the proteasome and possibly necessitating clearance by autophagy. And so this is one area that we're very excited about. We can also kind of return to Kyle's study and looking at how long-lived species turn over their proteins and manage their proteome. And we can look at their age selectivity as well as add a very unique species. So this is a degu, We actually have a colony of these on campus, and they are a rodent that naturally develops amyloidosis with high penetrance. And so it is possible that if we were to do this study, that the degus could be an outlier, for example, and that would be very cool. A lot of people have questioned if they can birth date their own specific proteins of interest, and of course you can, and we've done this. So we actually do have one case that we're working on writing up now, where we have in short-lived species with that fast turnover, this protein seems to be very quickly degraded. But in longer-lived species, where we have longer-lived versions of this protein, we actually see that it achieves kind of a hyper-modified state that actually confers a kind of a new function to this protein in specifically long-lived species. And we would, its proteome birth dating is very akin to protein birth dating, if you will. A last very exciting, more technical aspect is, again, that at the end of this birth dating scheme, each cell actually contains all of the information of the experiment. And so in this way, if one could get around some technical challenges, we could actually do single cell proteomics on these and evaluate the heterogeneity of protein turnover between single cells. And this would, proteome birth dating would be very good in this regard. And so with that, I'd like to conclude by kind of sharing this image that is representative of our method development, and kind of, this was from my thesis. So these were where I was focused in using the methodology across species in terms of global proteostasis and in terms of our specific targets. But I'm sharing it now just to kind of hopefully spur some encouragement. And we love open science. I'm very happy to be here. Thank you for listening to me. And I would love to work with people or have other people take this methodology and study their favorite science of interest and make some exciting findings. So with that, I'd like to thank you all for being here. Oh, well, I'd like to thank my labs, specifically Maggie and Sarah and Kyle, as well as some funding sources and the URMC MSTP.
Well, thank you, Michael. That's very exciting technology. And thank you for sharing all the things that are in preparation. I look forward to reading about all these. Please, if you have any questions, add them in the Q&A section or in the chat. I can start with one question and add up on the question. So, Michael, how easily can it be applied to different cell types, tissues, or organisms, especially considering possibly the variability in uptake of these amino acids? And building up on the question, I'm curious if you tried young versus old cells, senescent cells, or even talking about long-lived species, if you are would be interested in looking in centenarian people cells versus, because they have a clear advantage for longevity.
Absolutely. Yeah, that's a really good question and really exciting question. So the way we did these experiments for these purposes was like in primary human fibroblasts that are very well characterized primary cell laboratory line. And so we really tried to maintain everything as quote unquote normal or basal as possible. And so we haven't actually we've done some skin cells of a couple of other species, but within humans, we haven't really explored. And I'm not sure like our labs would necessarily explore. And so you bring up a very, very exciting kind of low-hanging fruit, which is we only looked in skin cells. So we could definitely take fibroblasts or kind of parenchyma cells from other organs like the liver and the brain, specifically like neurons or glia would be very exciting to look at. the human cells that we looked at are primary foreskin cells, so they were taken from a very healthy baby. We have not gotten into, for example, whether that could change with age, whether we took cells from a 20-year-old versus a 60-year-old or an 80-year-old or someone with AD. These are all really good and kind of low-hanging fruit questions that are begging for someone, a young scientist, to take over. And then in terms of like more biological context, there's also, so we did this on quiescent cells. So an exciting area could be to look at dividing cells, specifically kind of cancerous cells and other diseases like that.
Do you think it's applicable for in vivo? No,
In theory, it is.
in theory, yeah.
Yeah. So it would get expensive pretty fast. So there is for like Silac, there's Silam where you can Silac label a real mouse. The issue, so it would be that it already is expensive, but then we would have to like, or one would have to kind of custom produce the chow, and I would imagine that that would be pretty expensive. The alternative, though, and something that I've explored is using, I mean, at the lowest level, like organoids or iPSCs or something like this, but actually something like brain slice culture, for example, specifically in the degus, like if we had a degu brain slice versus a mouse brain slice, and could we birth date those? I think that would be super, super cool.
Yeah, that would be super cool. I have a question for the technique. So you have a succession, the sequential addition of the new code amino acids, and you have 4 days and 2 days. And how did you choose this
Yeah,
time? This time?
that's a super good question. I'm trying to go back to the slide just so we can, oh, there we go. So the reason we chose this time scheme, or rather the rationale originally was we were going to design a logarithmically decreasing timescale such that we can capture the greatest range and diversity of protein half-lives that we could. And we did some like math and playing with our equations to see kind of what that would be, and this seemed like a pretty good one. However, one could change this and tailor it to their specific protein or pathway of interest. So for example, that targeted case study I was talking about, that's a very short-lived protein normally. And so for that, we actually did a 6-hour, 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 6-hour time course. Similarly, kind of when we were in revisions, people brought up the idea that like super ancient, super long-lived proteins could really be like problematic in disease specifically. And to that end, you could adjust the timescale the other way, and you could really extend kind of your range. There are also, and we're working on this too, but there are more lysines available. You could, in theory, keep adding more labels. And there's a young grad student in the lab who I think was going to try for 11 labels at some point. I don't know how that's going.
That's very exciting to expand. I'm trying to read the questions in the chat. So from Vaishnavi, so if supposedly there were a similar experiment in vivo, hypothetically, how does the data presented here could correlate in terms of accuracy? So yeah.
Do in vivo.
Or more organic. Let's say, is your question about how can you compare data from say individual cells to maybe organoids, or . Right.
I
Sorry,
can,
right,
yeah,
yeah.
that's okay. I can speculate. Yeah, so I mean, hypothetically, doing the experiments in vivo would provide a lot of information and would certainly be more physiologically and biologically accurate. Um, you also you would also kind of get into the situation where when people have done this in the past with like dynamic SILAC, you can really get at how different organs have different turnover contexts. So, for example, I think the brain is a very slowly turning over organ in terms of their proteins, whereas the liver is kind of very quick. Um, and so, looking at organ to organ diversity in that case would be very interesting. Um, yeah, particularly in the kind of circumstances of disease. Um, I would say that like organoids and iPSCs and kind of differentiation protocols would also go a long way in achieving that physiological relevance in general. And then you get into the debate of organoids as a whole, and we won't touch that one.
Thank you, Michael. We are at 9 a.m. Time, so I want to be mindful of Michael's time and everyone's time. If you have any questions, you can post them on the bioprotocol, the protocol web page. And when Michael will have the opportunity, will have some time, he can reply to these questions. But it's super exciting. Thank you for sharing all this
Thank you
work.
for having me.
And yeah, it's great to have another option than DSI-like.
Yeah.